In 1901, a German psychiatrist named Alois Alzheimer examined a 51-year-old patient named August Deter, who was experiencing memory loss, paranoia, and confusion so severe she could no longer function. After her death 5 years later, Alzheimer examined her brain under a microscope and found strange plaques and tangled fibers nobody had systematically documented before. That was over 120 years ago. We have had a name for this disease for more than a century, more research funding devoted to it than almost any other disease on Earth. And still, today, there is no drug that stops it, reverses it, or comes close to what most people would call a cure. So what's actually going on? Is this a funding problem, a research incompetence problem, or is Alzheimer's a fundamentally different kind of problem than the ones medicine has already solved? The honest answer is closer to the third option, and understanding why requires understanding something most people have never had explained clearly. For the better part of 30 years, the entire field may have been organized around the wrong central idea. Here's the theory that has dominated Alzheimer's research since the early 1990s, called the amyloid hypothesis. The brain of a person with Alzheimer's accumulates sticky clumps of a protein fragment called amyloid beta, forming plaques between neurons. The hypothesis proposed that these plaques are the primary cause of the disease, triggering a cascade of inflammation and neuron death that produces the memory loss and cognitive collapse we recognize as Alzheimer's. It's a clean, testable idea. Remove the plaques, and you should slow or stop the disease. Billions of dollars in research funding, and nearly every major drug candidate developed for Alzheimer's over 3 decades were built on this single premise. There was just one problem. Drug after drug that successfully cleared amyloid plaques from patients' brains failed to meaningfully improve their symptoms. Trial after trial, across multiple pharmaceutical companies, produced the same disappointing pattern. The plaques went away, and the dementia continued anyway. By the early 2010s, some researchers were quietly asking an uncomfortable question. What if amyloid plaques aren't the cause of Alzheimer's at all, but a byproduct, a symptom of something else going wrong, the same way a fever is a symptom of infection rather than the infection itself? In 2022, that quiet skepticism turned into an open scandal. A neuroscientist at Vanderbilt University named Matthew Schrag flagged apparent image manipulation in a landmark 2006 paper published in Nature by researcher Sylvain Lesnay. A study that had identified a specific amyloid protein called a beta times 56 and claimed direct evidence that it caused memory impairment in rats. That single paper had been cited more than 2,300 times, making it one of the most influential Alzheimer's studies of the last 2 decades. And it had helped funnel hundreds of millions of dollars in NIH funding specifically toward amyloid research every single year. In 2024, after a lengthy investigation, the paper was formally retracted. Every author agreed the images had been altered, except Lesnay himself. This doesn't mean the entire amyloid hypothesis is fabricated, and most researchers in the field are careful to say so. But it does mean that for nearly 2 decades, one of the theory's most cited pieces of supporting evidence was built on doctored data, and that the sheer dominance of this single idea may have quietly starved research into other possible causes. Competing theories about brain inflammation, vascular damage, tau protein tangles, and immune system dysfunction that had far less funding and far less attention because amyloid had already claimed the spotlight. Which brings us to why Alzheimer's is such a uniquely difficult problem to solve even when researchers know exactly what they're looking at. The first obstacle is physical, and it's one that has nothing to do with which theory is correct. Your brain is protected by something called the blood-brain barrier, a dense layer of tightly packed cells lining every blood vessel inside your skull, designed specifically to keep foreign substances, including most drugs, out of brain tissue. It evolved to protect the brain from toxins and pathogens circulating in the blood. It is exceptionally good at its job, which is precisely the problem. Most drug molecules large enough to meaningfully interact with amyloid plaques or tau tangles are also too large to cross that barrier in useful concentrations. Getting a drug from a syringe into a patient's bloodstream is comparatively easy. Getting that same drug through the blood-brain barrier and into functional contact with the actual disease is one of the hardest delivery problems in all of pharmacology. Even when a drug does cross that barrier and does what it's designed to do, a second obstacle appears. By the time most patients receive an Alzheimer's diagnosis, they have often lost significant numbers of neurons already, sometimes for a decade or more before symptoms become obvious enough to seek a diagnosis. Unlike liver cells or skin cells, mature neurons in most regions of the adult brain do not regenerate. A drug that perfectly stops all further amyloid accumulation, all further tau tangling, all further inflammation, starting today, cannot bring back the neurons and the specific memories and connections that have already been lost. This is fundamentally different from a disease like an infection, where killing the remaining pathogen allows the body to recover. In Alzheimer's, stopping the disease and reversing the damage are two entirely separate problems, and current treatments, even the newest ones, can only meaningfully address the first. The two most recent FDA-approved drugs, lecanemab and donanemab, both work by clearing amyloid plaques from the brain, and both have shown, in large clinical trials, a genuine but modest effect, slowing cognitive decline by roughly a quarter to a third over 18 months, compared to patients receiving a placebo. That is real, measurable progress. The first drugs in the disease's history to demonstrably alter its trajectory rather than just treating symptoms. It is also not a cure. Patients on these drugs still decline, just somewhat more slowly, and the drugs carry a serious side effect risk called ARIA, brain swelling and microhemorrhages, that requires regular MRI monitoring and has been linked to a small number of patient deaths during clinical trials. There's a structural reason progress has been this slow beyond the biology itself. Alzheimer's clinical trials are exceptionally expensive and exceptionally slow to run, because unlike a cancer drug, where you can often see whether a tumor shrinks within weeks, an Alzheimer's drug's effect on cognitive decline typically has to be measured over 18 months to several years, using cognitive testing that is inherently noisy and variable between patients. A single phase 3 Alzheimer's trial commonly costs several $100 million and takes the better part of a decade to complete, from first patient enrolled to final published results. Multiply that by the dozens of failed amyloid-targeting drug candidates over the past 30 years, several of which failed only after reaching expensive late-stage trials, and you start to see why pharmaceutical companies have historically avoided funding Alzheimer's research relative to diseases with faster, cheaper, and more reliable trial outcomes. Diagnosis itself remains a serious bottleneck too. For most of the disease's history, a definitive Alzheimer's diagnosis was only truly possible after death, through autopsy examination of plaques and tangles in brain tissue. Living patients were diagnosed based on symptoms and cognitive testing, often only after the disease had already progressed significantly. Newer blood tests measuring specific biomarkers, including a protein called p-tau217, are now showing real promise in detecting Alzheimer's-related changes years before obvious symptoms appear, which matters enormously, because every current treatment works better the earlier it started, precisely because the neurons lost before treatment begins cannot be recovered. But these blood tests are still being validated at scale, and most people showing early biomarker changes today will never have access to them through routine healthcare. Put all of this together, and you get a genuinely difficult picture. A disease first identified over a century ago, whose leading scientific theory was significantly shaped for 2 decades by data that turned out to be fraudulent, protected by one of the most effective biological barriers in the human body, causing damage that cannot be reversed once it happens, requiring trials that take the better part of a decade and hundreds of millions of dollars to even evaluate whether a drug works, in patients who are frequently diagnosed only after the most treatable window has already closed. None of that means the field is failing. The newest drugs are the first in history to slow the disease's actual biological progression rather than just its symptoms. Blood-based early detection is closing the diagnostic gap faster than almost anyone predicted a decade ago. And the amyloid scandal, painful as it was, forced a genuine widening of the field. More serious funding now flows toward tau, neuroinflammation, and vascular theories than at any point in the last 20 years. Precisely because researchers stopped assuming one explanation had to be the whole answer. The honest answer to why we can't cure Alzheimer's yet is that we spent 30 years partly organized around a theory that turned out to be built, in part, on manipulated evidence, while fighting a disease protected by a barrier evolution designed specifically to keep help out. Causing damage our own biology has no mechanism to reverse. That's not a story about failure. It's a story about how long it takes to recover from chasing the wrong lead, and about a field that is now, for the first time in decades, actually looking everywhere else it should have been looking all along.